Good afternoon and good morning, everyone. Welcome to Swiss Korean Science Club Skeletal Muscle Disease Caused by Mutations in SDIM1. I'm Jian Lee, Junior Project Manager at Science and Technology Office at the Embassy of Switzerland in Seoul. We are part of the Swiss Network, the outpost aimed at connecting Switzerland and the world in education, research, and innovation. Our monthly Swiss Korean Science Club highlights and promotes the cooperation between the two countries in diverse fields of studies. This digital version of Science Club, which we started last year along with the COVID outbreak, is actually giving us new opportunities to be connected, at least virtually, with our partners in Switzerland and any other places around the world. So it is our great pleasure to welcome you all here today. Today's Science Club will start soon with two speakers from University of Geneva and Yonsei University, Wonju. Their research initiated with the advising professor of Professor Kyu-sang Kim in 2006 and is lasting until now with the Postdoctoral Exchange Scholarship funded by the National Research Foundation of Korea. The webinar will be under the moderation of Professor Klaas Wolheim from the Department of Cell Physiology and Metabolism, University Medical Center. I truly hope this Swiss Korean Science Club will offer every one of you a great time to learn about the latest research trends in Switzerland and in Korea. Also, I wish our Science Club opens up new opportunities for potential exchanges and joint cooperation between the two countries in the nearest future for the next participants of the postdoctoral exchange scholarship. Please use the Q&A function for the questions if you have any, and use the chat function for technical problems if you face any issues or difficulties. We will help you as soon as possible. Thank you very much for your attention. And now the floor is yours, Professor Klaas Wolheim.
Can you hear me? I have started my video.
Yes, we can hear you well. Okay. Thank you.
So my name is Klaas Wolheim. Welcome to this session. I start with a historic introduction. Since 15 years, Korea and Switzerland is collaborating through the links between Yonsei University at Wonju and Geneva University in Switzerland. It started with then assistant professor, now full professor and chairman of the Department of Physiology in Wonju, Kyu-sang Park, who came to visit my lab in Switzerland and Professor Nicolas Demorex's lab in Switzerland. So, and then we have had a number of scientific exchanges with visits of Professor Cha and Professor Lee in Geneva, and Nicolas Demorex and I have been visiting Yonsei both in Seoul and in Wonju to give lectures. And there have been a number of scientific, not only collaborations, but that have ended up with highly cited scientific papers. And this is we are all grateful to the research, to the Korean research funding agency that has made this, the National Korean Research Agency that has made this possible over the years. And I think Nicolas will come back to that. So I think that I wanted to say one more general thing. Oh, my video is not on? Is the video on now?
It is on.
Good. Sorry for this. So I wanted to also say one thing, that the topic that will be treated today is an aspect of how calcium is handled in cells, and in this particular issue in the muscle cell. And calcium is a universal messenger molecule, a triggering molecule in many, many cell functions, which Nicolas will come back to. But today we are dealing with a severe aspect of calcium dysfunction in a fairly rare, luckily enough, fairly rare muscle disease, but where the mechanism has been studied by Professor Kim when she was in Geneva, and in Professor Demorex's laboratory. So maybe just one word on the two speakers. Nicolas Demorex is professor of physiology and cell physiology in Geneva. He's also vice dean of the faculty and has many other feathers in his hat. And he has concentrated on the role of calcium in cell activation, and he studies different cells, including muscle, white blood cells, which are involved in the immune defense. And Professor Ji-hee Kim is research professor in Wonju at Yonsei University, and she's also interested in how calcium works in health and disease. So with this introduction, I should like to hand over to Nicolas Demorex, please.
Thank you very much, class. Can you all hear me?
Yes, we hear you well. Thank you.
Thank you, thank you. So I'm going to share my screen now. There you go. Do you see the screen?
Yes, good to go.
Okay, perfect. Thank you for the introduction, class. And thank you very much, the Science Club, for the invitation. And I'm very happy to see that Kyu-sung is in the audience. So I see the opportunity to thank you, Kyu-sung and Professor Shah, Professor Park and Professor Shah, for maintaining this cooperation alive over the years. It has been 15 years since we first met, and we had several opportunities to work together in Switzerland and in Korea, and it's really a pleasure. And I have to stress that it is really the exchange of people that maintain this collaboration alive. So it was a pleasure to have you, Ji-hee, and to see you again. I'm going to talk about calcium signaling in health and diseases, with a focus on muscle, because this is the topic that Ji-hee chose to study here. And when people talk about calcium, usually they think, it's for the bone. And this should not be forgotten that, yes, indeed, you need calcium to build a good bone mass. And this is especially important in childhood. So you need sufficient dietary calcium to build a strong bone mass that you will keep for your whole adult life. But then you also need calcium for your other organs to function, and importantly for your muscle to move. Without the calcium, you will not be moving. And this notion dates back to this person, Sidney Ringer, the British physiologist who discovered the importance of calcium in muscle contraction. Ringer is mostly known for devising the solution, the perfusion solution that you see at the left here. This is a substitute for blood, and this has saved countless lives. So I think he should be awarded a Nobel Prize for this invention, because if you just count the number of people saved by this invention, it's probably more than penicillin. So the interesting thing is that in the course of his work, he was trying to find what is in the blood in the 19th century. So he started with distilled water and then adding back different compounds, elements, ions, to try to match the blood composition. And at some point, he did a mistake, he or his assistant. So you can see in the text, which I quote here. And this is from the original paper. You see, this is a digital object identifier that links you to a paper which was published in the Journal of Physiology in 1883. And you can still read the text. So he said, I discovered that the saline solution which I had used had not been prepared with distilled water, but with pipe water. So his technician just used the pipe water. And then he found out, okay, this water, the pipe water from the London New River Water Company, contains some inorganic substances. So he had to see what was in there to recreate the right solution. And the observation was that with the pipe water, the heart of the frog was beating, but when he replaced that with distilled water, the beating stopped. And this experiment was reproduced. So you see in the right panel, you see a trace, and this one is actually from 1994. It's a paper by David Eisner. And what they did, they used this time a mammalian muscular preparation, and they made Ringer solution. You see here the two solutions, the one that was made with pipe water, they got to the same water supply in London, and the one that was made with distilled water. And you see when you replace the pipe water with distilled water, beating stops, the muscle cannot contract. Okay, so Ringer quickly found out that the magic substance was calcium, and that you needed calcium to generate signals inside the cells for this muscular contraction. And it's not only the muscle, because you also observed back in the 19th century that you need calcium. And you see here this calcium elevation. You need calcium for the very first event in life, which is the fertilization of the egg. So this is an oocyte from fish. And I play this again. You see that when the spermatozoid enters the egg, there is this calcium wave which propagates through the whole cell. And then there is this reaction, this global reaction, which is this zona pellucida, which is generated here, that prevents further fertilization. So this is ubiquitous. All the animal life will share this signaling circuit. And we know a lot now about how these calcium signals are generated, mostly through the invention of another man whom I would like to acknowledge, and this is Fluorescence, who unfortunately passed away 5 years ago, got a Nobel Prize in chemistry in 2008, sorry. He got it for his application of the green fluorescent protein, which he did not actually discover the green fluorescent protein, the GFP, but he made all the mutations that rendered it fluorescent at different wavelengths. And then based on that, he generated tools that can be used to measure calcium. Before this discovery that awarded him the Nobel Prize, he had designed fluorescent chemical probes that can be used to measure calcium. I'm going to show two examples. So the first one is a movie of cardiac cell. It's a precursor embryonic cardiac myocytes. And you see that when you just plate these cells and load them with this calcium dye, you can see a lot of activity. And this change in fluorescence intensity is proportional to the calcium concentration. So you can see, for instance, that you have global waves that propagate through the cells, but you also have local elevation that you can resolve at a specific location. And it is the encoding of this signal that generate a coordinated response. And with the genetic calcium indicator, you can now measure the signal in the whole organ. And this is a heart from a mouse, which is then expressing this fluorescent protein. And you see again that the color is proportional to the calcium concentration, and you see the elevation in calcium that coincides with the beating movement of this organ, the whole organ. And on the right, you have the same preparation, but with a much higher resolution, and you can now see the propagation of the calcium wave between the different cardiac cavities. So we know a lot about calcium signal, how they propagate, how they are encoded. One question remains is why did nature choose the calcium ion for this signal? And this is a complicated question to answer the why. We should always avoid the why question. But in that case, there is an obvious link, is that nature actually chose the phosphate as energy currency. You probably know that biochemical energy is stored in the form of ATP. which is a phosphate, and then you also have a lot of phosphate on the genetic material. So the cell is very rich in phosphate. And you probably know also that if you have calcium and phosphate together, that precipitates, so it will make CH-ROK. And to avoid that, what the cell devised, and this is very early in evolution, the cell devised systems to extrude the calcium ions. And these are calcium ATPase. These are energetic system. They would consume energy to remove the calcium ion, to push it outside. And the result of that is that the cell is now living in its cytosol with a very low calcium concentration in an environment which is very rich in calcium. There is a lot outside and very little inside. The gradient is about 10,000-fold. This creates condition where you can just open a door to calcium, a calcium channel, and then the ion will flow, and that would generate one of the waves that I show. This is obviously an efficient system, but it's quite dangerous because you can easily flood the cells with calcium. So the cell has also devised internal calcium stores. This is mostly the endoplasmic reticulum. They are equipped with a pump, a calcium pump, known as CH-ROK ATPase, and this accumulates calcium inside these stores. And with that, you can now release inside the cells at specific location. You have a system to release calcium ions exactly where it is needed. And using this system, the cells can encode calcium signals in the following way. You can generate signals here around secretory granules, and then you induce secretion of insulin, for instance. You can generate signals around calcium or potassium channels, and then you manipulate the cellular excitability. You reduce the heart rate, for instance. Calcium signals around mitochondria, you boost metabolism, and calcium signals around the nucleus or that are linked to a pathway that causes gene activation. Then you can drive cellular proliferation. And this is the signals that are linked to the pathway that we study with GE. And this pathway is also interesting in its historical perspective because it was conceived 20 years. The notion that it will work, the store-operated calcium entry, the notion is 25 years old, 25 years before discovery of the molecules. A model was proposed by which activation of the plasma membrane channels would be coupled to the emptying of the intracellular calcium pool, meaning that when the pool is empty, you have released everything that was stored here. When this is empty, this channel here opens, and this will allow the refilling of the stores and the generation of the signal. So this concept was formulated by Jim Putney in 1986, and the molecules were discovered 20 years later. This is a more modern version of the so-called store-operated calcium entry, where you have the signals that causes the emptying of the store, and when the store is empty, you have this question mark here. This store talks to this calcium channel. And we have here an important drug, which is tapsicagin, which is a drug that blocks this calcium pump that accumulates the calcium in the store. If you block this pump, you empty the store, you activate the pathway. And through this discovery, the molecules could be identified with the use of this protocol. So these two protocols we use a lot in the lab: fluorescence microscopy, the calcium probe, this Fluora probe, application of tapsicagin to release the calcium from the stores, and then you activate your store-operated calcium entry. When you re-add calcium to the cell, you have this huge calcium elevation. So this is the signal that we measure, or alternatively, we measure the entry of another ion, manganese, that will enter the cell and decrease the fluorescence. The other approach, which is also very useful in the field, is to directly measure the electric activity of the channel. Calcium is a cation. When it enters the cells, it generates a current. So when you record the cellular current, the current across the cell membrane, you can here induce the activation of this store-operated entry channel. either by this agonist or by depleting the store. And then you have the electrophysiological signature of this channel, which has this peculiar shape that basically highlights the fact that this channel is extremely selective for calcium. Pretty much only calcium goes through. So now the molecules, there are two, the STIM and the OI, and they were both identified by genetic screen. So here is just an example from one of the 3 papers that discovered the STIM protein in 2005. Genetic screen was based on this assay that I described. To deplete the store, you readd calcium and you measure the amplitude of this response. And here are the original data from the screen. These 2 genes, STIM1, STIM2, decrease the amplitude of this response. They code for a protein that is this shape and is assembled as a diamond. And it is in the endoplasmic reticulum membrane with a domain here, globular domain inside, that binds calcium. And that when calcium is bound, maintain the protein in this globular closed conformation, shielding these CAD-SO domain that are the channel activating domain. And when the calcium decreases here, the protein here extends. There is a zipping mechanism that basically will extend and expose those domains. The other important molecule is the channel itself. Its name is ORI. In humans, there are 3 isoforms, and it was also identified based on this screen using this calcium readdition protocol in cells treated with tachycargine. But this time they used, at least one study used genetic linkage, because in a family of patients suffering from severe combined immunodeficiency, they had observed that this patient 11, was lacking altogether the calcium elevation. And then they analyzed the whole family, and they could find in the parents that they had 50% reduction here in the amplitude, while the healthy people had a response that was double. And through the genetic linkage, they couldn't identify the gene and found out that it's a channel. And this is one of the latest version derived from the structure of this channel. It's a pretty long transmembrane domain with an opening here, hydrophobic domain. And then, very interestingly, here inside, there is another region which is charged, very negatively charged in here. And here is one of the recent view from the structure. It's a cryo-EM structure of this channel, which assembles as a hexamer, so 6 subunits will assemble. And here you see from the outside the entry, and these residues are negatively charged, would bind the calcium ions, and from the inside it's the opposite, they would bind the anions, and together that would ensure this very high selectivity for calcium. So the interesting point in this system is that to work, these 2 molecules have to find each other. And of course, you have the sensor, so STIM will know when the ER is depleted from calcium. When this happens, it will unfold, as I said, expose these domains, and these domains will enable the STIM proteins that would then form multimers. It will assemble into big assembly of STIM oligomers. It will bind to negatively charged plasma membrane lipids, and through this CAD domain, it will bind here the C terminus of the ORI channel, and that would induce the channel opening. And once the channel is open, calcium enters here and goes straight into the store. So they have to find each other, and for that, well, both have to move, both proteins have to move in the cell. And they have, in this process, they generate this very peculiar structure, which we observe by electron microscopy, and we term the cortical ear. So this is actually an ear cistern which has been elongated, and we found that STIM is actually contributing into this elongation process. And these membranes will be very close on each other, and they are enriched in STIM protein, as you can see here with these little dots They are gold label antibodies that signal the position of the STIM protein. And they make these contacts where you can observe a very dense structure. We believe that these are the sites of interaction between the STIM and the ORI. And here is another example. You see the endoplasmic reticulum is this structure which is decorated with ribosomes. And at this stage, there is a constriction, and the structure becomes thinner and gets apposed very close to the plasma membrane. So to generate that, the cell, when you activate, when you deplete the air, the STIM molecule, basically what happens during this unfolding and oligomer formation, it would move inside the cell and it will bind the membrane. And you can observe that by a fluorescence microscopy. And here we use a technique which is known as TIRF, total internal reflection fluorescence microscopy, where you see a very thin layer of the cellular membrane. And if I play this movie, you see here the dots are STIM molecules. And when we treat, in this case, this fibroblast with an agonist, after removing the calcium, you see that the STIM molecules accumulate in these dots, these cluster or these puncta. And when we re-add calcium, it disappears. So this is completely reversible, this process of cluster formation. Okay, the molecule assembles near the plasma membrane, as depicted in the sketch here in the top left panel. And what is interesting is when we do the same experiment, but this time in a cell which is myoblast, meaning it's a cell derived from a human biopsies, and you isolate the precursor of the muscle. And when you put them in culture, you can generate cells that would then contract. So you make what is called a myotube. And when this myoblast becomes a myotube, and you look at the behavior of the STIM protein, what we observe is that there is again formation of cluster, but in this case, there is more, there is an architecture, and you see that they form these longitudinal stripes. And we believe that STIM has a role in the making of the muscular triads, the sites where the muscle activity is controlled by calcium. And GE, which will present next her findings, will tell you more about that. Now, briefly to close, of course, if this store-operated entry pathway is ubiquitous, you can expect that there are some problems if it's not functioning properly. And this slide here highlights pretty much all the organs where a defect had been identified that is linked to defect in STIM or ORI. The two main systems are the immune systems. That's how it was identified in the first place. If you don't have the STIM ORI functioning well, you have severe combined immunodeficiency. So the main problem is in T cells, T lymphocytes. And the second is the muscle. The skeletal muscle cannot develop properly without the store-operated entry. And this is what happens in human. Loss of function is associated with muscle weakness, increased fatigue, and fiber atrophy. And this starts very early in life. On the other hand, the gain of function, meaning too much STIM or too much ORI, will also cause progressive muscle weakness. But there is a peculiar feature here that you will find these tubular aggregates forming inside the muscular fibers. And this usually happens later in life, this syndrome of again associated with Stimon gain of function. So I'm done. I hope I did not overrun too much. Just want to acknowledge people who are now in the lab, and the past member, Monica Bula, in particular, initiated the story that Jihi will present, and Amado, who's on top here, supervised also this story, together with Maud Frieden, who is now professor in the University of Geneva, who supervised the electrophysiology. So thank you, Jihi. I'll let you present your study on TAM, and I'm going to stop the screen sharing.
Thank you, Nick. Now the floor is yours, Jihi.
So I start?
Yes, please.
Yeah. Thank you, Jian and Professor Klaas and Nick for the introduction. Hello, everyone. I thank you all for attending this Korea-Swiss science meeting. I'm Ji Kim and working as a research professor at Yonsei Wonju College of Medicine. I would like to thank Kyu-sang and Seung-gyu, my supervised professors in Korea, for giving me the chance to work on Nicholas's lab in University of Geneva. I started to bring you to tell a story between my supervised professors in Korea and Professor Klaas and Nick in University of Geneva. Then I'm going to tell you my science work about skeletal muscle disease caused by mutations in Stim1. This slide is about the collaboration of Yonsei University and the University of Geneva, from Professor Kyu-sang, Park, Professor Nicholas, and Professor Klaas. Kyu-sang's research interest was mitochondria and calcium signaling. But at that time, there are few studies and no experts about mitochondria in Korea. To know how to approach mitochondria research as a basic research aspect, he was searching the lab where are leading this research area over the world. He found Nicholas and Klaas in papers who are top-class scholars in mitochondria research. Thus, Kyu-sang sent an email to them, and those two professors generously allowed him to study in their lab. During 3 years of effort, he successfully proved a very important role in mitochondrial morphology in insulin-secreting cell. In my case, I entered graduate school in 2010, and Professor Park was my committee member of my PhD. With Professor Park and Professor Klaas's support, I applied to the internship program in Professor Nicholas's lab. During 3 months, I conducted experiments about calcium channel gene related to skeletal muscle disease in his lab. After the internship program, I wanted to continue to study calcium signaling in skeletal muscle, so I applied for Postdoctoral Fellowship Program after 2 years and got a chance again to study in Geneva. So next slide, I would like to introduce how I got into this Postdoctoral Fellowship Program and what I have achieved during the time. The Postdoc Fellowship Program is supported by the National Research Foundation of Korea called NRF. It gives an opportunity to study abroad with scholarships for PhD within 7 years postdoctoral degree. For the application, you should prepare some documents, including a research proposal, as you can see here. In my case, based on the previous internship program, Monica, who was a previous lab member in Nicholas's lab, transferred her project to me. I wrote the research proposal thanks to her data, and finally, I won this grant. If anybody interested in this grant, you can get more detailed information by following this link. Okay, now I introduce what I've done in Geneva. My project is to demonstrate the pathologic calcium signaling by Stim1 mutant causing tubular aggregate myopathy. As Nicholas mentioned earlier, Stim1 senses the level of calcium in the ER. When ER stores are full, intra and intramolecular interactions keep Stim1 in an inactive folded conformation. When the depletion of the stores, calcium dissociates from Stim1 and promotes the protein extension and activation. This leads to the exposure of two important reasons, one binding to ORI1 channel and one binding to the plasma membrane. Activated Stim1 binds to ORI1 channel, aggregate them in clusters and open them. Calcium enters through ORI1 channel, and we call this store-operate calcium entry. Right side, you can see overexpressed fluorescence. So fluorescence tags Stim1 and ORI1 in cells. As you can see in this video, we can observe ORI1 and Stim1 form bright clusters on the cell membrane over time. The ER calcium was depleted in the middle of the experiment by applying a pharmacological inhibitor. So what happens to mutated Stim1 in skeletal muscle, and how does it regulate ORI1 by mutated Stim1? To find the answer, firstly, we should know the structure of Stim1 protein. The upper one is a normal Stim1 protein, which is composed of total 685 amino acids. The below one is a Stim1 mutated at 484 position, which forms a shorter length than normal Stim1. The I changes to R at 484 position, so we call this mutant I484R. I484R is composed of different amino acid sequence from normal Stim1 and between 484 and 503. It could make a change of 3D structure of the protein, and moreover, the mutated Stim1 might lose some functions in the domains after 484 position. This mutated gene is one of the genes found in tubular aggregate myopathy patients. He is 42-year-old man suffering from tubular aggregate myopathy. He has progressive muscle weaknesses in all four limbs and back muscles. And the strange and aggregated tubular shapes are confirmed in skeletal muscle biopsy, and it appears red in staining named here Gomori, or dark in NADH-TR. A muscle disease that shows their tubular aggregations on skeletal muscle biopsy is diagnosed tubular aggregate myopathy. Mutations in tubular aggregate myopathy studied so far lead to an excessive increase in calcium, resulting in tubular aggregation in muscle. So, does Stim1 I484R mutation also induce increased calcium? So this project aims to investigate how Stim1 mutant alters the ORI1 channel functions. Firstly, I confirmed Stim1 and ORI1 clustering, as you can see in this video. The upper two are normal Stim1 and ORI1, which are redistributed as bright clusters after calcium depletion. On the contrary, below two videos, the Stim1 mutant already forms Stim1 and ORI1's clustering from the beginning of the experiment in the condition with the calcium store field. What does it mean? It means that this mutant Stim1 activated independent with the calcium depletion. I also confirmed it in human myoblast cells. Olivier is a technician in Professor Mudd's lab who is an expert in skeletal muscle and electrophysiology. And for my experiment, he kindly had provided human myoblasts from human donors. In muscle cells, the size of a mutant Stim1 cluster is bigger than normal Stim1. Even more, they distribute irregularly and are moving for unknown reason. Then, do the preformed clusters impact cytosolic calcium levels? So next, I checked store-operated calcium entry and basal calcium level. You see here, a higher calcium value reflects a high calcium level. This gap reflects as a basal calcium flux, and later increase is SOP entry. As you see, the mutant has a high higher basal calcium levels as well as a high calcium, high SOK entry. Oh, sorry, its slide isn't stops. Oh, I should reopen the slide, sorry.
If your PowerPoint doesn't work, I could share the screen for you.
Wait a second, it opened.
Okay, perfect.
Okay, here.
Yes, good.
Additionally, we confirmed the basal calcium level in 5 human donors in the condition with exogenously overexpressed normal Stimon or mutant Stimon. The mutated Stimon significantly increased the basal calcium influx. So these data suggest that Stim1 I404R elevates basal calcium entry. Now, I wanted to further investigate the channel properties of ORI1 regulated by the mutant. To do so, electrophysiology is the best way to evaluate the function of an ion channel. The currents are generated by ions. The current through ORI1 and Stim1 is named ICREC. In terms of Stim1, I404R lost some domains associated with the calcium-dependent inactivation domain. The mutant may defect this function. Calcium-dependent inactivation is followed by activated channels to become inactivated to prevent excessive calcium influx, and this functions as a negative feedback. In cell co-expressing ORI1 together with the normal Stim1 or mutant Stim1, the density of the calcium current by the mutant Stim1 is significantly high, bigger than the one mediated by the normal Stim1, confirming the overactivity of the ORI1 channel by Stim1 mutant. Now, if you see the below graph related to calcium-dependent inactivation or CDI, normal Stim1 shows inactivation after generating the current by channel opening. The currents are decreasing over time because this CDI function. What about the mutant? Stim1 mutant does not inactivate it. So the channel gets prolonged its open state. Thus, this result suggests that calcium influx through Stim1 mutant mediated ORI1 is a result of a prolonged channel activation by loss of inactivation. Lastly, I checked the interaction between EB1 protein and Stim1 mutant. EB1 is a protein that is bound to Stim1 at resting state and constitute a trafficking mechanism restricting Stim1 targeting to ERPM junction. Normal EB1 binding Stim1 has been known to dynamically tracks microtubules. After store depletion, EB1 Stimon binding affinity is decreased and Stimon goes to the plasma membrane to make clusters, as I mentioned earlier. I bring again the protein structure of mutant Stimon to show that it loses the EB1 binding region composed with the TRIP. So we hypothesize that if there is no Stimon EB1 binding, Stimon mutant can move to the plasma membrane even when ER calcium store is full. So it is possible that Stimon proteins are accumulated into ERPM junction at resting state. So now I monitor this Stimon dynamic movement looking at the comet by taking photos every 1 second in a short time, like 30 seconds. In here, Stimon rapidly appears on cortical ER and disappears out of focus to deeper ER. So we call this comet assay. Visually, we can see the Stimon comet in wild type. However, the comet for Stimon was not detected in the mutant. Therefore, I conclude that a defective in binding with EB1 of Stimon to microtubules enhances recruit of Stimon into cortical ER near to plasma membrane. So this is a graphic summary of this STEM mutant, I484R. A resting state, wild-type Stimon is located in the ER and dynamically moves along the cortical ER or deeper ER. And EB1 is contacted Stimon between the ER and microtubule. EB1 bindings plays a role to delay Stimon translocation to ERPM junction during ER calcium depletion and prevent excess calcium store of calcium entry and ER calcium overload. However, due to TEM mutant doesn't have EB1 binding domain, so I484R leads to bring recruited on the ERPM junction to open ORI1 channel, continuously inducing excessive calcium. So I thank Nick to all the members of Nick's lab, especially I thank Amadou and previous lab member Monica directly advised me and helped me a lot about my project. Also, Professor Mudd advised and guided me about my patch clamp data. And all the members in Geneva always helped me in a kind and warm mood. I deeply appreciate Professor Park and Cha in Yonsei University, Wonju College of Medicine. And I would like to thank you all for listening my presentation. One thing more, are you curious of what Professor Park is up to now? Professor Park is now a leader in research about mitochondria in Korea. He established a mitochondria research center in Yonsei University, Wonju College of Medicine with the support from the NRF. Professor Nicholas and class still have collaboration on the basic science research with us, and I express my respect to their challenge and endless passion. With this, I finish my presentation. Thank you for your attention.
Thank you, Jihi. Now I invite class again for the Q&A session, and both Nick and Jihi, you can turn your camera on, please.
Mm-hmm.
If you have any questions, you can please use the Q&A function to leave your questions for the speakers, and we will address them for you.
Okay. Can you hear me?
Yes.
So thank you very much for these very clear presentations and for making a complex research area accessible to a general public. You did very well, Nicolas and Jihi. So we have some questions, and I think we should start with the questions to the last talk. And the first question to Jihi would be, since you have discovered the mechanism of how the gain of function mutation of STIM1 compromises muscle function, the question arises, if these patients that carry the mutation have wild type, which is normal STIM1, expressed in the muscle cell together with the mutant STIM1, and have the ratio between the normal STIM1 and the mutant STIM1 will determine the muscle function, so the phenotype of the patient. Could you dwell on that a little bit, Jihi? And Nicolas is welcome to join in as well.
I would like to answer first, Nick. Yeah, that's a great question, but unfortunately, I don't know the ratio of a wild type or mutant in skeletal muscle in 10 patients. I think to compare those expression levels, we might need to check the protein level of Stimon, normal Stimon, or mutant Stimon by Western blot in the biopsy sample. As you said, if wild-type Stim1 is more expressed than the mutant Stim1, it could make the disease progression slower because normal Stim1 functions are normally. However, I would like to say that more important thing is that the previous studies makes us think the ratio of STIM1 and ORI1 expression is critical to regulatory the channel function. It means that STIM1 regulates calcium influx depending on ORI1 expression.
Okay, thank you.
I could add a bit on that topic. I mean, it is known that the expression of both the STIM and the ORI is a biallelic, meaning that you have your two chromosomes and the gene on each chromosome in one allele, and they are both expressed. So this patient, they have one mutated and one normal. And the question, how much is true, it's not been measured in the muscle, but you can sort of anticipate that it's equal amounts. It's likely to be equal amount of the mutated and the wild type. And you can make the parallel to the situation with the SCID patients. And that's how the ORI channel was discovered. The patient who's heterozygous for the mutation, he lacks the gene on both because, both parents carry the gene. He had zero, he does have a zero stop at entry. The parent has 50% from normal. So you translate that to gain of function. If this patient would be homozygous, the gain of function should be worse. You can anticipate that. I think there is certainly a dose effect here. Also, all the mutations are not equal. Some gain of function are much more pronounced than others. You have some mutation that renders the channel always active. In that case, you just send some STIM molecule to the location where it activates the channel, and then you have more flux, but it's not as bad as having the channel always turned on.
Okay, thank you.
Thank you. And another question would, of course, be, since this is a human disease, is there any idea about a drug that could help these, medication that could help these poor patients, which have a gain of function mutation in STIM1, which of course also extends the question to the immune deficiency syndrome?
Yes, that's a very critical point. So far, this question is unsolved, so we should try to find a way. STIM1 and or ORI1 gene is not the only one gene that leads to TEM. There are other genes such as Caterin1, which is a calcium binding protein found as a TEM gene. So generally speaking, it has been known that calcium overload causes and accelerate muscle disease, but we don't know other specific pathophysiological mechanisms yet. So we can try gene therapy or calcium channel blockers in animal model first, and we can apply, after we understand the disease mechanism, we could apply the knowledge to develop the therapy, I think.
Have any non-selective calcium channel blockers been used to block ORI in the patient or in animal models?
In the
non-selective ion channel blockers.
Yes, there have been actually clinical trials for acute pancreatitis, and they used a so-called specific ORI channel blocker, but the truth is that there is no specificity, at least between the ORI isoforms and probably beyond, if it will also impact the TRP channels, this inhibitor. So, okay, if it's a very severe disease, then you can sort of forget about the off-side effect, immune modulation, etc. But for those patients to improve muscle function, I think we need to have something more specific than that.
Thank you. So then I turn to other general questions, which more addresses the Nicolas presentation. What makes the STIM signaling system so specific to other calcium signaling systems in the cell?
Well, I mean, it's unique in the way that, you know, it's a ligand-gated channel, but the ligand is not only intracellular, but it is on an intracellular membrane. So to have the system, you need to bridge two membranes. You have to bring an organelle to the cell plasma membrane. And this is really unique. And I think it serves the purpose, because the point here is to refill the organelle with calcium. So by bridging the membrane, then you can sort of generate a system where you bypass, you know, the calcium goes straight from outside to inside the endoplasmic reticulum without perturbing the cytosol. So this is unique in this respect.
May I add something to this, which is for the general public? The calcium signaling in a cell is unique because of the compartmentalization of calcium. So I always, and Nicolas introduced this, I always link or make the picture of a leaking boat. You have a rowing boat that leaks in water, and you have to have a shovel to shovel out the water. So the water is calcium, and it leaks into the cell, but it is used as a signal, and it needs to be kept low in the cell cytosol and the water of the cell, but it's high inside the organelle, the ER, and that makes calcium the signaling molecule in the cell. So you shovel out, that's the energy, that's ATP that is used to pump calcium out of the boat or out of the cell in this case, and calcium is pumped into the ER, shovel into the stores, which could be a reservoir in the boat. So you need to keep the boat with very, very low water, otherwise it sinks. That's the cell. Sorry, let's move on. What are the important questions? Sorry, Nicolas.
I noticed that Q-Sung has raised his hand, so you can hand him you can ask him for his question at some point.
Can he? So what is his? Hi, Q-Sung. Why don't you ask your question orally?
Well, actually, yeah, first of all, I just want to say my gratitude and say hello to both of you. And it's very nice to meet you at this kind of official meeting with very great meeting. And I would like to say here with officially that both of you are my mentors, and I have learned a lot, not only science, but also whole life and important things, essential things I learned from you. And I just want to give a academic question to Nicolas. And I was very impressed the pattern of lecture, the presentation is very kind to the general audience, and it's good to know the role of calcium and the calcium in the different compartments. So my question is, store-operated calcium entry is important for calcium provision to the ER and the cells. So is there many, I just want to know whether the store-operated calcium entry has important role in autophagy and lysosomal degradation and its function, because lysosome is a compartment that need calcium supply from ER or outside the cells.
Okay, good points. I mean, I'm not an expert in lysosome calcium signaling. From what I know, they have their own ATPase for refilling, so it's not a tachycardia sensitive pump. And they are usually coupled to the release of lysosomal calcium or acidic store is usually triggered by calcium release from the endoplasmic reticulum. So it's a kind of calcium-induced calcium release, if you want. That's the way, at least I see the system. So in that sense, yes, it would be coupled to store-operated entry, because if you don't have an ER calcium to trigger this, then you lose also this signaling. Then you have other second messenger like NADP that can mobilize these stores, and those would be independent on the store-operated entry, and they usually mediate very fast signal. At least in the T lymphocyte, it's a very like millisecond range calcium signaling circuit. But again, without competent ER, an endoplasmic reticulum replete with calcium, I don't think you could engage the signaling circuit. So yes, they will all depend on the SOCI, and actually the SCID patient sort of proved the points, because yeah, they would have normal NADP signaling, but without the ORI channel, it's just dead.
Can I just add for the general notion or comprehension that lysosomes are scavenger organelles in the cell that recycle, for instance, when the cell needs energy, they recycle, they degrade proteins and release amino acids to the cell to be reused. They also degrade macromolecules that are introduced from the external of the cell. So they have a very important organelle function, which again is regulated by calcium. Sorry, we are drawing over time.
Yes, so I think it will be, due to the time constraint, I think it will be the time to move on to the conclusion or wrapping up. So thank you all for your time and contribution, and also thank you to the audience for staying with us until the very end of our webinar, which is the last science club of this year. Thank you all, and we will have the recording on our website, so stay tuned. And for any updates on our office's activities or about the news from Switzerland and Korea, please follow us on LinkedIn. So all the speakers, please turn on your camera and smile at the camera so that we can take a quick screenshot of you.
Okay.
Perfect. And thank you all, and have a great rest of the day. Bye-bye.
Stay safe, all of you.
Thank you, Nick and class, and all of you. Bye.
A good day or good evening in Korea.
Yes,
Good
thank you.
evening.
You too.
Bye-bye.
Have a nice day. Yes.
Good morning.
Bye.
Bye-bye. Bye,
Bye.
Kisang.
Yeah, bye, class. Thank you.